a perfect opportunity to give you an update on what Autifony have been doing. Been working on Kv3 channels for some time, and I'll give you a perspective on, say, the direction of travel for this very interesting set of targets. In terms of the talk outline, I'll just take a slide in a moment to introduce you more to the company, and then get into the actual channels of interest. So a little bit about the Kv3 channels, why we're interested in them, and then the rest of the terminology outlined on this slide will become clearer as I run through the presentation. So Autifony Therapeutics is very much an ion channel modulator company, founded over 10 years ago as a spin-out from GSK. Obviously, in the early days, as many of you in the audience will know, a very small company and very focused effort on what was Kv3 channels only at that point. The company has grown markedly over the years. We're now 35 people, but always, so this is really crucial, supported by work with a wide range of international collaborators and contract research organizations as well. We certainly couldn't do what we do alone. In terms of the team, though, we do have extensive experience in the ion channel space, as you would imagine. People like myself worked in pharma for many years before for biotech, and worked from early stage idea generation, new targets, obviously, in terms of what we actually choose to work on, but also taking those all the way through the clinic and actually launching these, getting these approved as new medicines as well. So the initial focus of the company has matured very nicely. Autifony is now well recognized as having a lead position on the development of Kv3 modulators. We've taken these into clinical trials, developed all of the translational and other tools needed to really understand the effects of our compounds in the clinical situation, and that includes development of experience then in CNS therapeutics, but not just broad spectrum indications, but rare or orphan indications as well. And then I've already had a few conversations with people here about what else we're doing. I won't be presenting on this today, but part of my role at Autifony, one of the things I really have been keen and passionate to do is to expand our early stage pipeline. That's a more diverse array of targets, and that speaks really to the fact that we're not just choosing near family members to what we're already good at and what we're working on, very much driven by human genetic data, insights into disease, choosing the best targets that we can really make a difference on in terms of our particular experience. And that sees us going into ligand-gated ion channels, as well as the voltage-gated channels I'm going to talk about today, and also looking at organelle biology, and in particular lysosomal channels, which are, say, of particular interest. We're also working on neurovascular channels, and some of our programs, as mentioned here in terms of the company logos, are already partnered. So we already have an established collaboration with Boehringer Ingelheim, and one which was announced with Jazz Pharmaceuticals last year. But others within the company are also wholly owned by us, and will be the subject, I'm sure, of future partnerships and investment as well. So, putting Kv3 channels in context then. We obviously are all familiar with these sort of dendrogram representations of either part or all of the ion channel superfamily. It's why we're here. I'll be talking about the target labeled with the blue arrow here, Kv3, but obviously over the years this is a really rich source of ideas and talk titles for this particular conference. There are obviously other ion channels not captured in this particular representation. Kv3 channels then themselves, as captured in this schematic, you can see are part of the 6TM tetrameric channels, so individual subunits encoded by genes KCNC1 to 4. So there are 4 different Kv3 channels. They're all high voltage activated, meaning that they only really open at really pronounced depolarized potentials. And that really leads to their particular role in action potential repolarization. So they only really kick in when cells are actually firing action potentials, and they are predominantly expressed in neuronal cells. And in particular, and I may come back to this particular example, I'm sure, later as well, parvalbumin-positive interneurons, or PVINs, GABAergic inhibitory neurons, they're particularly highly expressing of Kv3 channels. So there are a number of ion channel medicines mentioned across the bottom, one from the last talk here. Obviously, it's a very successful target class in terms of development of new therapies in that respect. But currently, there are no approved medicines that are known to selectively target Kv3 channels. In terms of their expression, just a simple neuronal schematic. Kv3 channels can be expressed in dendritic locations, axon initial segment, nodally, or at the point where they can control neurotransmitter release. They're not necessarily in these positions on all cells that express, all neurons that express these particular channels, but that's part of the fun and the complexity of working on Kv3. They can certainly have a biological impact in each and every one of these situations. But what we typically associate them with is this impact on action potential firing. So in the absence of Kv3 influence, we might have an action potential half-width of this sort of scale, and with Kv3 present doing its thing, it will dramatically shorten the action potential, very short half-width, very fast repolarization, enabling that cell to fire again in a very short time frame, so to actually enable high-frequency firing. Looking at the biophysics of these channels, so I said there are 4 different family members. They sort of group 2 plus 2, the Kv3.1 and 3.2, which actually turn out to be the main targets of the compounds we've developed, have this more stable current over time, but all of these are actually activating rapidly and deactivating rapidly. But with the 3.3 and 3.4, which is somewhat different, this N-type inactivation actually leads to a very different profile of the currents. In terms of our modulators acting on Kv3.1 or 3.2, this is what we see. By influencing the biophysics of these channels, they shift the voltage dependence of activation to the left, so these conductance curves, and that leads to a greater, or is representative of an increased open channel probability of these channels at more and more hyperpolarized potentials. This is an example, obviously, of one compound working at reasonably low concentrations and shifting this GV curve. And in doing so, if we start then to look at neuronal populations, this is actually an experiment from a situation where there's cognitive dysfunction mediated or introduced via NMDA receptor antagonism. Particular neurons will show reduced firing rates, and these can be enhanced in the presence of a Kv3 PAM. So again, here showing a shift in the rate of action potential firing that these cells are able to maintain. So more on this later as well in a different disease context. So we've been working on Kv3 modulators for some time, as I alluded to. We've recently brought together quite a few different strands of investigation into this one publication, combining work from cytoretic mutagenesis, chimeras, and cryo-EM, and also molecular modeling studies as well. And it's very satisfying to actually finally, for sure, understand where the compounds, this time it's another relative of AUT201, AUT5, are actually binding on the Kv3 channel. And it's very satisfying to see that this binding site, and here's the compound shown here in this cleft, really does interact with a part of the protein that we've had interest in for some time, this extended turret region, which is a rather unique feature of Kv3 channels. And here it is on a more typical sort of topology map of the channels. So this extra bit of sequence here, highlighted here as well, between S5 and the pore helix, is very much a component of Kv3 channels and really not present in other potassium channels. This turns out to be, see, the binding pocket of our compounds, our positive allosteric modulators. But this turret region, which undergoes a sort of induced fit over the top of the compound, turns out to be really crucial in mediating the actual allosteric effect of these compounds on gating, promoting this increase in opening, this open channel probability that I mentioned. So there's a quick run through of the background of the understanding of, or identification and understanding of the the pharmacology and binding of our compounds. Let me now segue into some of the actual disease relevance for Kv3 channels and where we're actually going clinically with these particular molecules. So the first acronym, PME, progressive myoclonic epilepsy. A very important study published in 2015, basically disclosing the first causal mutation in the Kv3.1 protein, or in the KCNC1 gene, which gave rise to this particular condition. It's, as you'll understand, pretty dramatic in terms of its effects. The mutation occurs in the S4 region, or the voltage sensor domain. One of the crucial arginines is mutated to a histidine. So many of you will appreciate that that's a pretty, in most cases, severe mutation, amino acid swap to occur, and perhaps no surprise that this gives rise to a non-functional channel. That's when it's expressed alone as a sort of homomer then of the mutant channels. But in all cases, patients who carry this mutation have one wild-type copy and one mutant copy, so their cells will have wild-type channels as well. So although the actual mutation is fully dysfunctional, when we actually look at the heteromeric situation, if we look at cells which are expressing both wild-type and mutant channel, you can see there's a large downregulation in the Kv3 current available. It's clearly a loss-of-function mutation, but interestingly, there is likely to be a proportion of Kv3 channels which remain functional in cells, which therefore could be recruited, could be upregulated by use of an appropriate pharmacological agent. And that really is the basis of the approach and the attraction then of a precision medicine approach, which could be taken for these patients with this particular disease-causing mutation. We're interested in obviously addressing what might be the symptoms that these patients experience by directly restoring Kv3 function at that time. But also, as I'll come to and revisit later in the talk, obviously these patients have lifelong dysfunction of Kv3 channels, and therefore there is opportunity perhaps to actually positively affect the circuits which are dysfunctional in these individuals and actually have a disease-modifying effect as well. In terms of the other acronyms, MEEC is myoclonic epilepsy and ataxia associated with a potassium channel mutation, and EPM7 is just another classification for the type of PME that these patients are experiencing. So how do we build confidence that we can treat the sort of symptoms and disease course that these patients experience? Firstly, worth looking at exactly what they experience. Just in brief, key points about this disease: it has an onset in teenage years typically. Myoclonus is one of the most debilitating aspects of the disease, so a real loss of muscle control associated particularly with tremor when individuals try to voluntarily move their muscles. So it results in ataxia, complete loss of coordination and muscle control, and other aspects like a dysfunctional level of communication then as well, again from a lack of normal ability to use the muscles involved in speech. It's not life-threatening, but it is a severely debilitating disease which leaves people mostly wheelchair-bound by the time that they're at the end of their teenage years. So how do we build confidence that we can help these individuals treat this disease? We've actually been working with Ethan Goldberg in the US at CHOP to develop a mouse model, a transgenic model of this disease. So here we can introduce the very same mutation that was identified in all of those human subjects, introduce that into the Kv3.1 protein in mice, and then obviously build understanding as to whether that model actually phenocopies the human disease. It does. I'll come on to that in more detail in a moment, but just a first look in terms of some ex vivo studies, like I was saying with the impact on neuronal firing earlier. If we look at, in this case, cerebellar neurons from essentially these meek mice, we do see a truncation, a reduction in the firing frequency, so the red here compared with the black of the wild type. So these cells are much less able to fire a train of action potentials. If we introduce one of the compounds, AUT201, from 0.1 micromolar upwards, these are higher concentrations of the compound. In all cases, even the lowest concentration is sufficient to restore the firing of these neurons to more wild-type levels. And actually, interestingly, the wild-type ones seem to already be at a maximum, so we're attenuating the dysfunction, but we're not really affecting the normal physiology of these neurons. So that seems like an encouraging effect at the ex vivo or the cellular level. And then looking at behavioral studies, done a lot of work here. Apologies, this is a very sort of busy slide, but it allows me to make a number of points, perhaps while you absorb some of the data. We're looking at a number of different behavioral readouts in these meek mice. In the first instance, we're looking at hyperexcitability or the seizure susceptibility of these animals, and this is studied using a GABA antagonist, pentylene tetrazole, to induce a progressive kindling in these animals. Wild-type animals are in the dotted line here in black. Here is the clear higher susceptibility of the meek mice to this particular experimental paradigm. If we introduce AUT201, the same compound as just now, at either 5 mg per kg or 20 mg per kg orally, not only are we able to reverse that phenotype, but actually there's evidence of just a raw anticonvulsant effect in the wild-type animals as well. So seizure susceptibility seems to be addressed. If we look at rotarod as a measure of ataxia, there is an impairment in the animals. So I said the mice seem to phenocopy the human disease. And if we introduce, there's a trend at 5, if we introduce the compound, and at 20 mg 201, there's actually a attenuation of the ataxia phenotype in these mice as well. So seem to be able to address both of those key endpoints in the animals. This is another compound, AUT206. This compound also shows a similarly beneficial effect in the pentylene tetrazole kindling model. I include it because we also in this study looked at spontaneous seizures which do develop in these animals at older age. And again, in the presence of the compound, there was a clear effect in terms of anticonvulsant benefit against the seizures in these animals. This work has now been published in Cell Reports in collaboration with Ethan Goldberg. All of the data has been presented at epilepsy conferences in the last year or so. So what about essentially broader aspects of epilepsy? There's something else which has been building as a story in parallel to this work, is that there is actually, and we've had a number of talks today mention different rare diseases essentially which have been identified through gene sequencing. This is having an impact across the board for epilepsy, but a whole range of CNS indications clearly. For KV3.1, I'll come to KV3.2 in a minute, it's certainly allowed us to identify other CNS diseases, other epilepsy indications, which are also due to dysfunction, mainly loss of function in the KV3.1 protein in that channel. So beyond the MEEC or the PME I've been talking about, we have DEs, as have been mentioned a number of times today. We have other myoclonic epilepsies, and we even have intellectual disability aspects of developmental disorders as well, which are separate to epilepsy. For KV3.2, the studies actually support a much broader impact as well, not just the DEs and others I mentioned, but actually into generalized epilepsy, focal epilepsy, and other neurodevelopmental disorders such as autism as well. What you'll see, here are the primary papers supporting these headlines. What you'll see in this schematic that I put together is that the mutations which are occurring in 3.1, 3.2, and other actual members of this family are actually reasonably spread throughout this protein. So there's lots of things that can go wrong with these KV3 channels that can lend themselves to disease. So there's really a lot of work to do here to understand these channels and the relationship with pathology in detail. just a couple of last slides, as well as the genetic clues in terms of where we go for MEEC and these other epilepsy indications. This term, interneuronopathy, I mentioned KV3 channels are very heavily expressed in these parvalbumin-positive GABAergic interneurons. Dravet syndrome is actually quite a prevalent epilepsy disorder in which the PVINs are particularly hard hit, actually due to, in most cases, 70-80% of cases, due to mutations in the sodium channel, NAV1.1. What we've learned from studies on these animals in collaboration with Alex Rotenberg is that the compounds here, where we know there is, say, dysfunction in these interneurons, again, KV3 positive allosteric modulators can attenuate the seizures in these in a mouse model of this disease. So very encouraging data showing potential based on the ability to modulate this inhibitory network as well. And of interest, the animals were also studied in parallel in terms of looking at the expression of KV3 channels. And what we've learned is that KV3.1 is actually reduced in the animals, so not all go on to actually exhibit seizures at old age, but in the ones that do, the level of KV3 channels is actually markedly reduced. So there seems to be a dysfunction in the interneurons, seems to be a loss of KV3, which the KV3 PAMs can address in this context. What I mentioned about the disease-modifying effect, the way we think about the treatment paradigm that could develop here, as well as treating symptoms in patients at older age, addressing their seizures. If we are able to actually modify the neuronal dysfunction, upregulate the inhibitory networks, which are clearly impacted by some of these genetic mutations, and get in early, perhaps after first diagnosis, there is clearly a much greater opportunity to really affect the trajectory of the actual patient journey, affect the actual, attenuate the build-up of disease symptoms which will occur in these individuals due to the actual ongoing seizure burden and other dysfunction that is clearly present from birth. So getting in early with a KV3 positive allosteric modulator may actually be a really good opportunity. And then just to finish, this just shows, I won't go into this in detail, but this term interneuronopathy is being applied to a wider and growing range of CNS indications as well. So be it traumatic brain injury, Fragile X syndrome, Alzheimer's disease. There's clearly dysfunction in inhibitory networks for which new modulators such as the KV3 compounds could have significant benefit. And we hope to go on and explore this in the coming years. So it just remains me to thank everybody at Autifony who's contributed to this work over many, many years, and also a range of collaborators which I've mentioned as I went through this talk, but in particular the Covarrubias Lab, Goldberg Lab and Rotenberg Lab, whose data I've shown today. Thank you very much.
Thanks, Martin, for a great talk and a really cool subfamily of potassium channels I've been interested in for a long time as well. Do you have one quick question? Yes, the back.
Yes, so Martin, what do you think is actually... Martin, what do you think the mechanism is? Is it shortening action potentials, or actually, when you look at Dravet, is it the after hyperpolarization that's key? So increasing the size of after hyperpolarization.
So the way these channels affect the repolarization, it should actually be, that should be the predominant effect. So getting the actual cell ready to fire again. I think at high concentrations of the compounds, it is possible to actually modify the AHP, but in normal therapeutic concentrations that we're targeting, that shouldn't actually be altered.
Okay, thanks very much, Martin.